Hello viewers, I am Dr. N.T. Rafiq, Professor, Department of Agronomy, Assam Agricultural University, Jorhat. Through this video, I am going to demonstrate the standard procedures involved in seed selection, seed treatment and incubation of rice seeds for sowing in nursery as well as in the main field. These procedures, when properly followed, will facilitate higher yield of the rice crop and also reduce seed rate and also reduce the need for replanting or gap filling.
Good quality seed is the prerequisite for growing a healthy crop, which is obtained from healthy plants free from insect pest and disease infestation. A good sample of paddy seed should be dried up to 14% or less moisture, genetically pure, having high germination capacity of more than 80% and free from weed seeds, inert matter and pest infestation. Now, seed selection plays an important role in obtaining healthy, fully filled seeds suitable for sowing in the main field or nursery bed. Using ill-developed, partially filled, shriveled or broken seeds which contain less nutrient for developing into normal seedling, which can recover quickly from the transplanting shock and ultimately influence the crop yield by about 5 to even 20%. Only the very best seeds which are true to type of the selected variety, healthy, viable and clean with high germination percentage of more than 80% should be used for sowing. Seeds of higher specific gravity possess better germination and give rise to higher percentage of healthy seedlings which recover quickly from transplanting shock. Seed selection in wet condition is found to be more efficient than dry method. In wet method, the healthy seeds are selected by using common salt solution to a certain specific gravity which varies according to the species of rice. For example, a specific gravity of 1.08 is considered optimum for indica group of rice. The unfilled seeds float above the solution and the well-developed healthy seeds sink in the solution. For obtaining a salt solution with specific gravity of 1.08, we need to dissolve about 1.9 kg of common salt in 10 liters of water, which will give us about 19% strength of the solution. To test whether the solution concentration is right or not, use a fresh egg or a whole medium-sized tuber in the solution. If the egg or the tuber sinks, add little more salt till it floats. Pour the seed selected for nursery in the solution. Stir the seeds well in the solution. Allow it to settle down and discard the floating seeds. Select the sunken seeds which have settled down in the solution. The same salt solution can be reused to test another seed lot. Salt solution treated seeds need to be washed thoroughly. Seed treatment. Seed treatment is a term that describes the uses of specific products and specific techniques to improve the growth environment of the seedlings and young plants. It refers to the application of fungicide, insecticide or a combination of both to the seeds so as to disinfest for destruction of surface-borne organisms that have contaminated the seed surface but not infected the seed surface, and also to protect them from seed or soil-borne pathogenic organisms and storage insects. Seed treatment may be done by dressing, coating, pelleting with the products or soaking in solution.
After discarding the floated seeds, we have to select the healthy sunken seeds.
The selected seeds are then immersed in the solution. Here we are using carboxyn at the rate of 2 gram per liter per kg of seed.
We have taken 3 kg of seed, so 6 gram for 3 kg seeds and kept in the fungicide solution for any seed-borne pathogen for 24 hours. After 24 hours, we have to go for incubation of the seeds.
Seed soaking. Seed soaking means keeping the seeds under water for 12 to 24 hours to help the seeds absorb sufficient water and remove germination inhibitors present in the seeds for better, uniform and early germination. Incubation is the next step.
After completion of seed treatment for 24 hours, the treated seeds are kept for incubation. It is incubated to provide the required temperature by generation of heat for proper sprouting of the seeds, and it is kept for 36 to 72 hours depending upon the ambient temperature. For Kharif season, it may take about 36 hours, while for Boro season, it may take 72 hours. And this way, the seeds are incubated. And point to be remembered is never to use plastic bags. In an airtight condition, gunny bags should be used for covering the seed lot and kept for incubation for about 36 to 72 hours for proper sprouting to be sown in the main field or in the nursery bed.
Germination is very much important for seed quality. Higher the germination percentage, less is the seed requirement. We have tested the germination percentage of the seed which will be sown for nursery bed. So we have taken 50 number of seeds in the petri dish, and then we have tested the germination percentage. And after 2 to 3 days or in room temperature, only 46 number of seeds have germinated. So the germination percentage of the seeds tested is 92%.
Now let's have a recap. Selection, treatment and incubation of paddy seeds before sowing. Select healthy and bold seeds by preparing a salt solution, by dissolving common salt of 1.96 kg in 10 liters of water. Test the soil solution concentration with a fresh egg or a whole medium-sized potato tuber in the solution. If the egg or the tuber sinks, add more salt till it floats. Stir the seeds well in the solution, allow it to settle down and discard the floated seeds. Select the sunken seeds which have settled down in the solution. Salt solution treated seed needs to be washed thoroughly. Now the steps in seed treatment. After selection, seeds should be soaked directly in fungicidal solution for 24 hours. A fungicide solution is made by adding 2 grams of carboxyn per kg of seed per liter of water. 1 liter suspension is required for 1 kg of seed. After 24 hours of soaking, seeds should be kept for incubation. Next, the steps in seed incubation. Seeds are taken out from the soaking container. Excess water is allowed to drain. Seeds are placed on top of a platform in a shady area and tightly wrapped with gunny bags. Seeds are then incubated for 36 to 72 hours uninterrupted. Check the seeds after completion of incubation for uniform sprouting.
Hello viewers, I am Dr. Ranjan Krishna Sharma, Assistant Professor, Department of Agronomy, Assam Agricultural University. So I am going to present before you a video. This video is on the technology for raising rice nursery. Generally we go for 4 different types of rice nursery. One is wet bed nursery, another one is dry bed nursery, and third one is the mat nursery, and another one is the depot nursery. So in the state of Assam, we generally follow the wet bed nursery, and nowadays we are promoting the most scientific method of raising rice nursery, that is the mat type of nursery. So, in this video we will demonstrate the technology involved in raising a wet bed nursery and mat nursery. These two type of nurseries we will demonstrate in this video. So, all of we know that in transplanting method of rice cultivation, we need to raise the rice seedlings in a nursery bed, and later on we uproot the seedlings from the nursery and we transplant in the main field. So, for this process we need to raise healthy seedlings in nursery by following standard scientific methods. So, we will learn the steps involved in raising a wet bed nursery as well as a mat nursery, which is most scientific method for raising a rice nursery.
Rice is cultivated mainly by adopting two different methods of establishment. Number 1, transplanting method. Number 2, direct seeded method. The transplanting method of rice cultivation requires seedlings to be raised initially in a nursery bed. Generally, the nursery area required to provide seedlings for transplanting in 1 hectare is approximately 1/10 of the total farming area in wet bed nursery and dry bed nursery. The different types of rice nurseries are: number 1, wet bed nursery; number 2, dry bed nursery; number 3, depot nursery; and number 4 is mat nursery. So in this video we will discuss the wet bed nursery and mat nursery. In case of wet bed nursery, this method is suitable in areas with sufficient water supply. For transplanting in 1 hectare area, 40 to 45 kg of seeds is required. The farmers of Assam predominantly follow wet bed nursery for manual transplanting. The steps involved in wet bed nurseries are as follows. First one is seed selection, next seed treatment, number 3 incubation, and 4 is the raising of nursery. So now I am going to discuss the steps involved in raising of nursery. The first step is preparation of the seed bed. Land is thoroughly puddled and seed beds of 10 meter length and 1.25 meter breadth are prepared with 30 centimeter gap in between the beds. The height of the bed should be raised to 5 to 10 centimeter from the ground. However, the length of the bed may be according to the convenience of the grower. Next step is application of manures and fertilizers. In each seed bed of size 10 meter into 1.25 meter, 20 to 30 kg well-rotten cow dung or compost, 80 gram urea, 80 gram single superphosphate and 40 gram muriate of potash are to be applied and mixed well with the soil at the time of land preparation. Next is the seed rate. Well-germinated seeds are to be sown at the rate of 650 gram to 1 kg per bed depending on grain size. The beds should be covered during the initial 3 to 5 days with local materials like banana leaf, rice straw or with plastic seeds to protect from rain and bird. The cover should be removed after 4 to 5 days. The next step is the water management. Irrigation water should be applied in furrows to maintain saturated condition in the surface soil of the nursery bed. However, standing water to a depth of 2 to 3 cm should be maintained at least 2 to 3 days before uprooting. Next step is pest management. Insect pest, diseases and weeds should be controlled by adopting suitable pest management strategies. Now we are going to discuss about the mat nursery. Mechanical transplanting, that is using self-propelled rice transplanter or manually operated mechanical transplanter, requires seedlings raised in a mat type of nursery. A mat type of nursery is a nursery where rice seedlings are raised on a thin layer of soil and farmyard manure or compost mixture placed on a polythene sheet. The seedlings raised in this type of nursery resemble a tightly knitted mat at the time of uprooting. The polythene sheet prevents the seedlings' roots from penetrating to the underlying soil. This type of nursery is prerequisite for mechanical transplanting. The mat can be cut into desirable shapes to fit the trays of the transplanter. These seedlings can be used for manual transplanting too. The materials required for mat type of nursery are polythene sheet, wooden or iron frame, well pulverized and well sieved soil, farmyard manure, sprouted and treated paddy seeds. For preparation of the pre-sprouted seeds should be treated as per recommendation in a fungicide solution for 24 hours. Dimension of plot. The length of the bed should be 16 meter and the width should be 1 meter. 3 numbers of such beds are needed to prepare seedlings for 1 hectare main field. Frames are required, so removable frame is used to ensure uniform mat size. The dimension of the mat frame should be equal to the dimensions of the tray of the transplanting machine. Mechanical transplanter have inbuilt trays or seedling boxes. Preparation of the soil mixer. Sieve soil, particle size less than 4 to 5 mm, is mixed with farmyard manure in the ratio of 2:1. The FYM is also crushed to ensure homogeneous mixture. The mixture should be free from stone or any other foreign objects. Next step is the preparation of the seedling bed. Polythene sheets are placed on the selected plot of land. The frames are placed over the polythene sheet. The prepared soil and farmyard manure mixture is used to fill up the frame over the sheet to have a bed dimension of 100 cm into 1600 cm into 1.5 cm. Wooden mallet may be used to level as well as to provide mild compaction to the soil FYM mixture. Next step is sowing of sprouted seeds. Sprouted seeds are uniformly sown in the soil bed at the rate of 1 kg per meter square. Beds are sprinkled with water after sowing. Water management. Beds are covered with light jute bag or straw and water should be sprinkled on the bed for 4 days under covered condition so that soil never dries. The covers are removed after 4 days. The sprinkling of water may need to be continued up to 1 week. After about 1 week, seedlings would attain some height and water may be applied by flooding. The depth of water should be half of the seedling's height. Finally, uprooting of seedlings. The seedlings become ready for transplanting when its height becomes 15 cm with 3 to 4 leaves in about 15 to 20 days. The seedling mats are then cut into pieces to be used for transplanter. The advantages of mat nursery are it uses less area when compared to conventional nursery. Met nursery produces robust seedlings 18 to 20 cm tall in about 15 days. It minimum root damage as the seedlings are not uprooted and transplanting shock is reduced. And finally, labor requirement for nursery raising is significantly reduced.